Hello, this is Mr. Anderson, and these are the instructions for what these students are going to be doing for their work for science class for September 3rd and September 4th. What we're going to do is a gizmo. Now, most of the students have done a gizmo already. We did one about the triple beam balance last week. So the first place the kids need to go is to the portal. The portal is the best place to access anything that they need to sign into through Northwest ISD. The reason why is it signs you in automatically. So the gizmos are found here in the link that says Explore Learning Gizmos. We'll click there. Now we also on the portal want to open up Google Classroom because this is where our data sheet is going to be found. So we'll open up Google Classroom and we'll go to the correct class and we're going to click on classwork, and that's where we're going to find where it says Cells Gizmo Data Sheet. Let's just open this up. And that gives us a copy of that. Looks good. And let's go back here and check out what we're going to do on our gizmo. Now, everybody should have at least two gizmos listed. One should say triple beam balance. That's what we did earlier in the week. The new one will be the one that says cell types. So we want to click on cell types, and then we want to launch the gizmo and get it started. So this gizmo is practice using a microscope. We're going to look at different types of objects, mostly cells, to see what's in the cells so we can get an understanding of what all the cells have in common and essentially what makes up something that is living, because all living things have cells. So when you click on the gizmo, you'll notice not a whole lot going on. There's a girl sitting by a lake, there's a cow, there's some little animals and some grass and stuff. If you field over stuff, you will see things start to highlight. There you go. It's really hard to see where things are that are highlighting. And so for that reason, what you need to do is click where it says Show All Samples. We're going to click that. And that highlights everything, which is really, really nice. So with everything highlighted, we're going to go back to our data sheet. That's what we opened up from Google Classroom. Now the data sheet has some different cells or objects on the left: E. coli, red blood cells, it says it's in blood; euglena, which says it's in protists, that's important information; fungus, sand and silt, and human neuron. So we're going to look at all of those different samples. And what we're going to look for is to see if these have a nucleus, or cytoplasm, or a vacuole, or a cell membrane, or a cell wall, or a flagella, or an axon. And then we're going to determine, are these living things or not? So we're going to do a lot of work, but this tool is going to make it pretty efficient to do. So let's start with E. coli. We're going to investigate this. Okay, so we're going to go back to the gizmo, and we're going to find E. coli, and it's actually over here where this cow is. So we're going to click that, and when I click it, notice the cow shows up. It says a little bit about some stuff up here. And what I am actually interested in is the microscope, which is this little tab here in the middle. So once I have this highlighted, I'm going to click the microscope. And what that does is bring this gray screen up, and not a whole lot, because we need to zoom in on the microscope to see these little cells. Now remember, a microscope is used for zooming in or magnifying the sample, something that's very, very small, too small to see very well with the human eye. So what we're going to do for all of these samples, where it says magnification, we're going to go and we're going to click to the highest magnification. In this case, it's 2,500. For some of them, it's 400, but we're going to click to the highest one. And when we do that, we start to see a little bit of stuff here. So that's good, but we need to zoom in a little bit more, and we also need to clarify everything by focusing it. So what we're going to do is go to where it says Coarse Focus, and we're going to start moving this bar nice and slow. Let's move it to the right, see what happens. Not much. Let's move it to the left and see what happens. And as I move it to the left, let's see here. There we go, all the way to the right. Oh, I've got to focus all the way to the right. Now notice they're still a little bit blurry. That's okay, because once I've done my coarse focus and gotten it pretty good, then I'm going to use my fine focus. And I'm going to see if I can get it even clearer. I'm going to go to the right a little bit. Notice they get more and more clear, and then they get very clear. There we go. So I can see these little guys. These are what the E. coli look like if they're magnified 2,500 times. Now, I want to know what these little structures are. What I have to do is click Show Labels. So I'm going to click that. And look, there we go. I see all the stuff. It says cell wall, cell membrane, cytoplasm, flagellum or flagella, plus something or other, a nucleoid. Interesting. So I'm going to go back to my data sheet. And nucleus, is there a nucleus? If I look here, it says nucleoid. That does not say nucleus. So I'm going to say, no, I'm not going to check that box. How about cytoplasm? Let's see if there's cytoplasm. And there's cytoplasm. So I'm going to go back here. I'm going to click cytoplasm box. Okay. How about vacuole? I don't see a vacuole. No. Then I've got cell membrane and cell wall. And I look here, I got both a cell wall and a cell membrane. So I'll click both of those, cell membrane and cell wall. And then I see flagellum, and then it says flagella, because those are basically the same thing. So I'll click that. And then it's axon. Let's see if there's axon here. I don't see anything about an axon, so I'm going to leave that blank. And then I want to figure out, is this a living thing? So what we're going to do is click on this right-hand little tab that says Test for Life. We're going to run a test to see if this is a living thing. Click this right here and click the little control. And if it turns green, it is a living thing. So I can go ahead and check this box that it's a living thing. There we go. So what you're going to do is the same thing for each one of these samples. And you're going to find each one of these samples in the Landscape tab. And you can click on them and figure out where they are. Now, one thing I'll show you also, the red blood cell says in blood, euglena says in protist. If I click over here, blood is here, so I need to click this to see the red blood cell. Protists are here, so I need to click this to see the euglena. And just a little hint, when I click this and then go to the microscope and go to 400, let's focus it a little bit. Oops, there we go. I'll use fine focus, get it better. Notice that everything is kind of offset. I'm going to use these controls. This says move the stage left or right, so I'm going to move it this way. And then move the stage up or down, so I'm going to move it this way. Now I can see the euglena, so now I can answer the questions about the euglena. Okay, so that's what you're going to be doing. The goal is to get all of this completed, so then we can understand what these cells have in common and what they have that is different. And then we can figure out what makes a living thing a living thing. Now, if you have any questions, if you get stuck, the best thing to do is watch this video again, because these are the very, very clear instructions. Of course, you can always reach out to me, but this video will help you a lot. So don't let yourself get stuck. If you don't know what to do, just watch this. All right, good luck.